Hello and welcome back. We've got another great session coming up. It's entitled Evolving Vaccines, and that means that we've got a lot of interesting topics. I've got the pleasure of introducing the first two speakers. My name is Professor Robert Boy, and then I'll hand over to Professor John Litt, who will chair the second half and lead the discussion. So I have the pleasure of introducing our first speaker, Associate Professor Cameron Webb, who's been a mosquito aficionado for more than a century, 25 years. And he's done a lot in mosquito-borne disease research and management. He provides advice to local, state and federal government on mosquito control, and he provides advice to the public. I heard him only on the radio a short time ago. He's involved in public health interventions to reduce pest and public health threats to the community generally. I always enjoy his talks, and I look forward to hearing today's. Thank you so much, Cameron.
Thanks, Robert. That's a great pleasure to be joining you today. And I'm speaking to you from the traditional lands of the Wangal people in Western Sydney, and I pay respects to elders past, present, and emerging. As Professor Boy mentioned, I spend most of my summers sloshing around wetlands chasing mosquitoes, and today I'm probably going to talk to you more about the changing face of mosquito-borne diseases in Australia, and particularly New South Wales, where I'm based. It'll touch on some of the vaccine-preventable diseases, but unfortunately, for some of our worst mosquito-borne diseases, there aren't currently widespread available vaccines. So I'll touch on some of the dynamics of these diseases, how they circulate amongst mosquitoes and local wildlife, how you can protect yourself from mosquito bites and these diseases, and overall touch on some of the ways in which our cities and climate are changing that might result in the emergence of greater risks associated with mosquito-borne diseases. Mosquitoes are already considered the most dangerous animals on the planet. The pathogens they transmit kill almost a million people every year, but there's hundreds of millions of people who are infected with mosquito-borne pathogens, particularly malaria parasites. I'm not going to talk about those today. I'm going to concentrate on the arboviruses, but there are plenty of those to be concerned about and are spread by mosquitoes that are found right across the planet, including here in Australia. But mosquito-borne disease is also something that is regularly talked about when we think about the implications of our changing climate. You'll often see suggestions that we'll see more outbreaks of tropical mosquito-borne diseases when we start living in a warmer and wetter world, but it's a much more complicated equation to solve when we think about how a changing climate will influence local mosquito-borne disease risks. This is something I think about a lot here in Australia, and we have to understand not only how patterns of rainfall and temperature change, but particularly extreme weather events like flooding and drought, which can all impact local mosquito populations. You may not have ever thought about this before, but even coastal storm events, sea level rise can also change the complexity of mosquito populations that we're having to deal with. But it's more than just climate as well. And so the ways in which we build our cities, the way we design our cities to respond to these climate change risks will all influence mosquito populations as well. But overall, we're finding that mosquitoes are finding a way to dodge the ways that we've been killing them in the past. Insecticide resistance is something that we're seeing on the rise amongst some of the key mosquito species that are involved in the transmission of arboviruses. And if that continues, we're going to have to look for new ways to control mosquito populations. It's not just about the development of vaccines, it's about the development of other techniques that might involve the genetic modification of mosquitoes or the introduction of pathogens and parasites into mosquito populations that might change their life cycle ecology or ability to transmit these pathogens. There are thousands of mosquitoes across the planet, but there are a small number of those that play an important role in transmitting these pathogens that make us sick. But mosquitoes aren't like a dirty syringe that transfers droplets of infected blood from person to person. There's a complex biology involved in the ways in which mosquitoes become infected and become infective for some of these pathogens. If we can find a way to disrupt that mosquito biology, we can stop their ability to transmit some of these viruses. There's lots to talk about in that space. I won't spend a lot of that time today, but just to mention that there are a lot of different ways in which we need to assess the risk of mosquitoes and their management into the future. And this is really critical because these images that you're seeing on this screen now, these clouds of insecticide that we usually see accompanying stories about outbreaks of mosquito-borne disease, this really isn't a sustainable way to control mosquitoes. It's a really blunt instrument. And while it may have worked in limited areas in the past, we can't rely on these insecticides to use anymore. Many of the key vector species of concern are becoming resistant to these insecticides. And that not only impacts our ability to respond to outbreaks of mosquito-borne disease, but also limits the ability of authorities in Australia to respond to the introduction of exotic mosquitoes that might bring the capacity for outbreaks of things like dengue to Australia. And that's something I spend a lot of my time thinking about. So when we think about the arboviruses of great concern internationally, and particularly in the region that Australia is placed and countries where Australians may be visiting, yellow fever, Zika, and chikungunya virus are some of the mosquito-borne pathogens of great concern. These viruses are typically associated with tropical regions of the world, and they're spread by mosquitoes that are most commonly found in water-holding containers. You'll hear me talk about this more and more. Aedes species like Aedes aegypti and Aedes albopictus that are found in water-holding containers that are common through a lot of urban and metropolitan areas across tropical countries of the world. These viruses are intermittently quite active in many regions and only brings a risk that Australians are going to be infected by these pathogens when they travel, but infected travellers might then be a pathway for these pathogens to come back to Australia. And I think we need to be mindful about this, even for something like yellow fever, which does have a vaccine available. We've seen resurgent activity in Africa, and increasingly, we're mindful about that risk of reintroduction of yellow fever into countries in Asia where there are suitable mosquitoes that can transmit this virus. And we already know that international travel can be a pathway to introduction of these viruses into new regions. We saw the 2016 outbreak of Zika virus, which you may remember even threatened to cancel or reschedule the 2016 Olympic Games. It's funny to think about the parallels between Zika virus in 2016 and a lot of the discussions we had about mass gathering events due to COVID-19. Chikungunya virus itself, an outbreak in Central and Southern America in 2015, again highlighted how these viruses can move around. And so we need to be mindful about how these viruses may make their way to Australia with infected travellers in the future. But our big concern is dengue, of course, and the burden of disease in many countries surrounding Australia continues to grow. And it's interesting to see some of the commentary around the possible disruption to the health system amongst many of these countries that's been resulted from our need to respond to COVID-19 and what the unintended consequences may be to degrade the quality of the public health responses to dengue in many of these countries. And so I think we're going to continue to see dengue as the most important mosquito-borne arboviral pathogen in our region. Over 40% of the world's population is at risk of dengue. More than 100 countries have seen endemic activity, and there's estimated to be anywhere up to 400 million cases of disease every year. This is a disease of our cities, and that means that increasing urbanization around the world provides ideal conditions for these viruses. These viruses can cause severe disease and have incredible impact on local communities. We know there is a vaccine that is available for dengue, and I'm not going to talk about that in great detail today, mostly because this is not my area of expertise, but I would draw attention to some of the position statements the World Health Organization have made around this vaccine and where it's appropriately used for certain members of the community that have already had exposure to dengue in the past. But what I like to concentrate on here is the fact that the mosquitoes that transmit dengue viruses are usually those species like the Asian tiger mosquito, the yellow fever mosquito, that are found in water-holding containers in our backyards and urban areas. And this means that there is a great potential for these mosquitoes to be introduced to Australia. And they're being introduced from regions where Australians are moving to travel to. They're countries from which freight and aircraft and shipping are coming into Australia. And those populations of mosquitoes are demonstrating a high resistance to insecticides. And so when I work with the authorities in Australia, we're concerned that the mosquitoes that are coming into the country are resistant to insecticides. If they become established in our major metropolitan areas in Australia, there is a real risk that infective travellers coming back into Australia may trigger local outbreaks of dengue, as well as other pathogens like chikungunya and Zika viruses. And this is something we're seeing all the time now. There's reports from authorities from around Australia that these exotic mosquitoes are being detected at an ever-increasing rate. And we know from colleagues in North America and Europe that this is a pathway of introduction of these mosquitoes. So you may hear lots of discussion about climate change and how that's bringing tropical mosquito-borne diseases to new regions. But it's important to remember that it's much more complicated than that. And often before we can have the virus introduced with infected travelers, the environment needs to be seeded with these mosquitoes that are capable of transmitting these viruses. As we stand in New South Wales, there are no mosquitoes present that can currently transmit dengue, Zika, or chikungunya virus. But as we'll learn later in this presentation, there are mosquitoes that can spread a range of different other local and exotic mosquito-borne pathogens. But I am working with local authorities to build our capacity to respond to these exotic threats. For many years, we've concentrated on responding to exotic mosquitoes as being the major trigger for increasing the risk of some of these exotic mosquito-borne diseases. But as we learned from this summer, that's not necessarily the case. We've got to think more broadly when we think about future mosquito-borne disease risk. But it's important before we talk about that to talk about some of the local mosquito-borne pathogens. You've probably all heard about Ross River virus. It's responsible for the most mosquito-borne diseases reported annually, about 5,000 cases around the country. But we suspect that's just the tip of the iceberg, and many more cases of infection actually occur. Historically, we've generally thought about this as a rural disease, but we're seeing increasing number of cases around our cities. We're also seeing increasing number of seasons where record-breaking seasons for activity are occurring. Here in New South Wales and Southeast Queensland, we saw one of the major outbreaks of Ross River virus disease in 2020 that coincided actually with the COVID-19 pandemic. So a reminder that mosquitoes don't adhere to social distancing and lockdown to stop other infectious pathogens. Mosquitoes are still out there in the environment spreading these pathogens that can make us sick. Ross River virus won't kill you, but it can make you very sick and result in severely debilitating illness. Predicting outbreaks of Ross River virus can be incredibly difficult because of the wide range of mosquito species that are involved in that transmission. Whereas in Australia, we may only have 1 or 2 species that can transmit dengue virus, there are around 40 that have been implicated with the spread of Ross River virus. And it spreads the virus not necessarily directly from people to people, but also from animals in the wildlife. So when we're trying to green our cities to respond to climate change, what we're finding is that we're creating conditions that can actually predispose our cities and metropolitan areas for outbreaks of things like Ross River virus. And one of the key components there is the conservation and rehabilitation of our bushland and wetland areas that provides ideal conditions for our wildlife. In the case of Ross River virus, that's mostly macropods, such as kangaroos and wallabies, but the role in increasing populations of water birds in our cities as wetlands are being installed, rehabilitated, and enhanced for wildlife conservation is something that authorities need to be mindful of. And we got a good example of that in recent years, which I'll talk about shortly. So for the summer of 21-22, many of you will, like me, will think about that summer as being marked by incredible extreme weather events, and particularly flooding events in parts of New South Wales and Southeast Queensland. The predictions now is that we're heading into the 3rd consecutive year dominated by La Nina weather patterns. And that means excessive rainfall across many parts of the country. And it might come as no surprise to you that the more water we see, the more conditions are ideal for mosquito populations. And more mosquitoes mean the potential for more transmission of mosquito-borne disease. Now, when we see these excessively wet years, our mind often turns to one of the local, potentially fatal mosquito-borne pathogens, Mrna Valley encephalitis virus. Only about 1 in 500 people who are bitten by an infected mosquito will develop symptoms. There's no vaccine to it. But we have seen some major epidemics in the 1950s and the 1970s. And this occurred at times when we had excessive rainfall, above average rainfall. And it really kind of focuses my mind on the potential for mosquito-borne disease when we've experienced these previous summers of incredible flooding events where locals in these regions of inland New South Wales, Victoria, South Australia and Queensland talk about flooding that's unprecedented that they haven't seen since the 1970s. And of course, my mind is turned to Mrna Valley encephalitis virus. We saw some activity of the virus during the flooding in 2011, and with the rainfall that we had in the past summer, all of our minds were turning to the potential for Mrna Valley encephalitis virus. But something else happened. there was the discovery of widespread activity of Japanese encephalitis virus, which came as a shock to everybody. This was not something that we were expecting to see in temperate regions of Australia. Japanese encephalitis virus is one of the most important mosquito-borne diseases in Asia and the Western Pacific. It's estimated there's about 70,000 cases a year. Symptoms can be mild to severe, but it can be potentially fatal. Only about 1% of those that are infected go on to develop these potentially fatal and severe symptoms, but it is still a significant impact and a significant risk to Australia to have widespread activity of this virus. Interestingly, the transmission cycles are involved with water birds, pigs, and mosquitoes. So it's a little bit different to some of those other tropical mosquito-borne diseases that are mostly spread by mosquitoes found in water-holding containers. Japanese encephalitis virus is spread by mosquitoes associated with wetlands, rice paddies, and other freshwater type environments. It's also mostly spread by Culex mosquitoes, which are very different to those Aedes species like the Asian tiger mosquito or the yellow fever mosquito that are found in very different ecological niches. There is a safe and effective vaccine available, and even though we do provide recommendations to travellers that are going to endemic regions, my understanding is that that's rarely uptaken. And we've seen that in recent years with reports of Australian travellers coming back from Indonesia and Southeast Asia that have been exposed to Japanese encephalitis virus. But as I said, we never expected to see widespread activity of the virus in Australia. But in early 2020, we saw activity of Japanese encephalitis virus in a number of piggeries. And there's been evidence of activity in piggeries in South Australia, Victoria, New South Wales, and Queensland. We've also seen human disease, the virus detected in mosquitoes and sentinel chicken populations, sentinel chickens in New South Wales. And there's also evidence from the Northern Territory and other parts of Australia that there's activity of the virus in feral pig populations. In late February, early March, the Commonwealth declared a communicable disease incident of national significance. And we've since gone on to report 40 cases of human disease, and sadly, 5 deaths have been reported as well. And I think this is significant. So my perspective is on New South Wales, and I think it's really important for us dealing with the management of mosquitoes and surveillance of mosquitoes that for the first time since the 1970s, people have died in New South Wales because of a mosquito bite, and that's because of the arrival of Japanese encephalitis virus. There's now subsequent evidence that there's widespread activity of the virus. It's not just limited to piggeries. And the questions have always been asked, look, is this due to climate change? What role has climate change played in this outbreak of disease? And Japanese encephalitis virus poses a really interesting situation for us because it's not just about native animals that are playing a role in here. It's piggeries, it's feral pig populations. We need to take into account those populations. But importantly, we have to need to think about our native bird populations and how they're responding to climate change and may predispose us to Japanese encephalitis virus risk. Now, we know that there's been extensive laboratory and field work done since the detection of Japanese encephalitis virus in the Torres Strait during the 90s, together with a lot of the work that we've done on Murray Valley encephalitis virus across mainland Australia. We know a lot about the types of mosquitoes that are spreading these viruses and what role where they need to be targeted from our surveillance points of view. Culex annuli rostris, a mosquito found in freshwater environments widespread across Australia, is the species of greatest concern. And it comes as no surprise that the La Nina-dominated weather patterns have been ideal for this mosquito. There's been extensive flooding across many parts of inland regions of Australia. That's provided ideal conditions for mosquitoes, but also water birds, the main reservoir hosts for the virus. So with these increasing predictions of extreme weather events, this is something that we need to understand. These conditions have no doubt led to the cascade of the virus through mosquito and waterbird populations from northern areas of Australia, where the virus may have been introduced by migratory birds or wind-blown mosquitoes from Indonesia or Papua New Guinea, and has cascaded through the country into temperate regions of Australia. There's lots of techniques that we can employ to manage mosquitoes and their transmission, and together with government and industry, we've been developing strategies to combat this. But notwithstanding the availability of a safe and effective vaccine, personal protection measures to avoid mosquito bites are going to be critical. And thankfully, some of the advice that we share every summer will still apply to preventing the bites of mosquitoes that may transmit Japanese encephalitis virus. So using a topical insect repellent that contains DEET, picaridin, or oil of lemon eucalyptus will be effective. Applying it as a good even coat over all exposed areas of skin at dawn and dusk when mosquitoes are most active will help prevent mosquito bites. Now, unfortunately, this outbreak has not been a one-off, and we expect that with environmental conditions continuing to be conducive for mosquitoes and water birds, we should expect to see activity of Japanese encephalitis virus in the summer ahead. There's been some extensive Commonwealth government funding to this, and we hope that there's now going to be a national approach to surveillance of Japanese encephalitis virus. And together with cooperation through industry and government, we hopefully will be able to fill some of the gaps in our understanding about mosquitoes and wildlife, their role in transmission of Japanese encephalitis virus. So we hopefully, fingers crossed, won't see any more deaths resulting from this virus in the summer ahead. Thank you, Robert.
Brilliant, Cameron, as always. Really informative. And I've got a load of questions, but I'm not allowed to ask them because our next speaker is Dr. Simon Thatcher, who, like me, graduated from the University of Queensland in the 80s and trained at the Royal Brisbane, and I'm in Gold Coast and Mount Isa. So I've got to ask you lots of questions too, Simon, but I know you've done a lot of work on travel medicine. You're also a very busy practicing clinician. You've moved to the Gold Coast and you've been there now for a long time. And we're really pleased to have you speaking again. I remember when you spoke a few years ago at this same forum, at this same conference. It was great. So we look forward to today's presentation. Thank you very much, Dr. Simon Thatcher.
Travel vaccines. We're going to talk about travel vaccination in the age of resistance. I'm speaking to you from the Gold Coast. I respectfully acknowledge the people of the Agumba region, the traditional owners of the land I am speaking from, and pay my respects to elders past, present, and future. I have no particular conflicts of interest. I am a clinician, not a researcher, so my talk is very different. I'm not going to present lots of charts, so much to talk about what we are doing with our current travel patients. And yes, thank you for asking me back. I spoke 3 years ago. I'm very surprised to be back given the lack of my clinical expertise. And we'll start by talking about a hypothetical traveller. Now, I've de-identified this traveller, of course, changed his name, so I hope no one recognises him. We're going to call him Tim the Traveller. So Tim doesn't want any vaccines. He travels, he does whatever he likes. He decides to go to Laos, and this is what he sees. And he said, I want some of this. He did see us before he went, and he got some azithromycin, which is for emergency treatment of severe diarrhea, which he got. So he developed a fever and bloody diarrhea, and he took them and it didn't work. So as part of antibiotic stewardship, we try and discourage patients from taking antibiotics for any mild or moderate diarrhea. So it's got to be severe diarrhea. They've got to have like fever, blood with it. And fluoroquinolones no longer work, which is actually not too bad because of the risk of tendinitis and other infections, overgrowth in the bowel. So we give them a single dose of 100 milligrams of azithromycin. And why does it no longer work for E. coli with toxins is because we now have azithromycin resistance spreading throughout Southeast Asia. And there is no other oral antibiotic. There is a non-absorbable antibiotic called rifaximin, which is for non-ETEC, that is non-toxin producing E. coli, but it doesn't help with the toxin producing ones. And I say not too many people actually use that because that falls into the group of diarrhea that people can sort of put up with on their holidays. So he had his 3 days of severe diarrhea with blood and fever, and he came back and said, could I not have prevented that? Well, you could point out that eating whatever he likes is probably not a good idea. But there is one vaccine that is helpful, the oral cholera vaccine. So it is trademarked Dukoral in Australia. It produces antibodies to both the bacteria, but also the toxin. And that's the important bit because the cholera toxin is similar to the ETEC toxin. Of diarrhea in less developed countries, roughly 40% is caused by ETEC type toxins. And there's a lot of variability. It can be as low as 20, it can be as high as 60. But we encourage a lot of travellers to use it now because it's the only ones we can prevent. And bad diarrhea is the most expensive thing in preventing days of a trip. That is preventing you going and doing an activity. Coughs and colds are more common, but people actually continue to function with coughs and colds, continue to be able to do stuff. So bad diarrhea is a constant issue. And of course, it is becoming worse in the age of resistance. So you get 2 years of protection for cholera, but 6 months only for the ETEC toxin. if they do it every year, they have a booster. It is cost effective. That is, if you look at the days lost to travel, even though the vaccine is only partially effective, they will save more money than they will lose by doing it. It can be given to children. It's completely inert. That is, it is not absorbed into the system. And it is very sweet and a bit disgusting. And occasionally I've had travelers who said they just can't drink it, but it is otherwise well tolerated. And you can even try and force it down children with a bit of encouragement sometimes. So Tim comes back to us and he heads off to Bangladesh, and you see these lovely fields where they are growing all of their vegetables that you will be served in your salad. And he says, well, I've had my Gicorel this time, so I can safely eat all of those things without any great problems. But of course, the issue then becomes Salmonella typhoid. Typhoid is unfortunately, again, becoming increasingly resistant. There's very high resistance to third-generation cephalosporins, and again, increasing resistance to azithromycin, and there are really no other oral alternatives. Climate change, of course, doesn't just mean droughts, it also means floods and bigger floods, and that is increasing all forms of food contamination, because floods mean sewage runs into this food, so not just Salmonella typhoid, but also other species like cholera and Shigellas and Campylobacters and all that sort of stuff. So I had a patient from Egypt, and he was a young 25-year-old, and he actually physically saw them washing his salad in the Nile while he was on a felucca on a boat, and he promptly ate it. And he ended up with both Salmonella typhoid, a non-Salmonella typhoid species, a Shigella species, and also some undefined parasite. Ended up in hospital for 3 days and survived. Unfortunately, there was a medical student from Queensland several years ago who did an elective in India and died from typhoid fever. So 28-year-old healthy person, it is a big problem. So because of these issues, we follow, I use a website that is constantly updated, and it basically takes all the information from the CDC, and they give guidelines on what risk or whether vaccines are recommended effectively. So typhoid used to be in a lot of countries recommended for only adventurous eaters off the usual tourist routes, but now it's indicated in most travelers. So this includes countries like China or Turkey, where you normally wouldn't think, oh, do they really need typhoid just to go to China for a couple of weeks? Well, the answer used to be no, if they eat carefully, nice accommodation, but the answer has changed. And in fact, a lot more travelers should be doing typhoid vaccination. It is only 70% effective, and I actually don't know whether the medical student either had it and it didn't work or didn't have it. I would assume he probably had it. Oral typhoid vaccine is also works quite effectively, but is currently very hard to get because of supply issues. And like a lot of other medications, travel doctors are having problems getting vaccinations. The Gicorel I talked about, there is currently a shortage, although they tell us it will be back in a month or two. The type and the injectable one is available. The oral vaccine has an advantage in that it also protects against some of the paratyphoid species, which the injection does not. So if you have a choice and it's available, it is a slightly better choice. It is a live vaccine, though, as opposed to an inactivated vaccine for the injection. The big thing about typhoid is if you treat it really early, the fatality rate is very low. But if it's delayed, the fatality rate can increase quite dramatically because they develop quite a toxic septicemia. And the early symptoms are pretty innocuous, things like a fever, some mild abdominal pain, maybe a bit of bloody diarrhea, as you would expect from E. coli and things like that. Fecal testing is poor. Even blood culture has about a 50% failure rate. And bone marrow testing, which is far and away the best test, but is clearly very invasive and not available to most travelers. So effectively, you think you have to treat early if you expect it. So if you don't get better from something like azithromycin for ETEC, then those patients should be seeking further medical care and not waiting too long. In the United States, they've actually had resistant typhoid cases in non-travelers, which means they have transmission either between people or from their foods. The US is a weird place because they grow all of this cane everywhere and wheat, but they import all of their fruit and vegetables from Central America, and they constantly get outbreaks of Salmonella and E. coli and even hepatitis A, and then say, oh, I wonder why that's happening. But the grain industry is very well protected by lobby groups. It's an incredibly weird situation that a country that could grow so much fruit and vegetables just chooses not to, but I guess that is their choice. So what does Tim do next? He has his Typhon VI, and this time he says, I'm going to go and work in the slum areas of India for 12 months. This sounds like fun. Doesn't get too much diarrhea, but he comes home and then he develops a cough, and then he starts coughing up blood. And unfortunately, the testing shows he now has extensively drug-resistant TB. This is an increasing problem again because of the long-term administration of drugs to treat TB, which allows resistance to appear, and also that in countries where tuberculosis is very common, people often get better after a couple of weeks, feel really good, and simply stop the treatment, but that just allows resistance to breed. So first there was drug-resistant TB, and now we have extensively drug-resistant TB, which often requires 5 different medications for up to 2 years. The cost can be north of $1 million for whichever sort of public health system you have, which is one of the reasons that tuberculosis is causing more and more deaths in less developed countries, because they simply can't afford that. There are some schemes through the WHO to support the price of that, and also some of the philanthropic groups. But we certainly don't want a lot of cases of this in Australia because it's also dangerous to staff and everything like that. There was an MAGA report a few years ago, again, of a young man who he went through about 8 or 9 different medications, had masses of side effects, some of them extremely bad, like, renal failure and stuff. And so it is just not an easy treatment. And one of the hopes of people like myself is that we can develop a much better tuberculosis vaccine, riding on the back of all the research being done on mRNA vaccines and things like that, because governments are essentially subsidizing that vaccine research when the companies won't do it because there's no profit in developing new TB vaccines for poor countries because they can't afford to pay for it. So what does BCG do? So its main purpose, of course, is that it is given either at birth or soon after birth in less developed countries, and it can reduce death and hospitalization by about 75% in children under the age of 5 from systemic TB infection. That is, the TB doesn't stay in their lung, it goes to the rest of their body, and that is both very hard to treat and has a high fatality rate. It is still the most given vaccine in history, so nothing has yet overtaken it. There's over hundreds of millions of doses given every year at the moment. Not in Australia, though. So Queensland Health has stopped completely, and that is a problem for travellers or families who live in Australia but came from overseas who are clearly going to go back. They used to give those babies BCG, but it doesn't happen. And so now as travel clinics, we are doing BCG, often in older babies, though, or even young children, which is a nightmare because trying to hold a 2-year-old and give them a slow intradermal injection can be quite traumatic for everyone involved. The main new contraindication is the newer immunosuppressive medications, particularly TNF inhibitors. I have yet to see this, that if a mother has been on TNF inhibitors, you have to delay giving the baby BCG for 6 months. So it's an extra question we have to ask if we're going to do tuberculosis vaccine. So what about BCG in the older traveller? So what difference would it have made to Tim? So there have been a number of studies done in different countries, and the data, unfortunately, is a bit all over the place. But there is a chance that the vaccine will also stop TB getting into your lung, not the main purpose, which is stopping the developing of the extensive TB disease. So decrease in latent TB, that is TB in your lung, in people over 5 is thought to be somewhere in the 20 to 25%. And that goes up to the age of 30 at the moment because that's as far as we've researched it. It's a very safe vaccine provided the person is not immunosuppressed. And that's not a great amount, 20 or 25%. But if you compare that to getting extra drug-resistant tuberculosis, then in some travelers, we're going to have to start really thinking about recommending they get BCG. One of the problems with that is, of course, you have to do it quite a while before they travel. BCG is not a fast immunization. It's given intradermally. It takes months to produce its full effect. And so you can't really give it at the last minute because it won't be helping them for quite a long time. But, provide the person is particularly healthy, and if they're going to do a trip which puts them at substantial risk of tuberculosis, we import BCG because it's not available, of course. We're using a European variety. There's also Indian varieties you can import. A bit of paperwork involved, but it's possible to do. And I think more people are going to have to look at that. BCG is a really interesting thing, because it doesn't just protect against tuberculosis, it also stimulates your immune system and protects against a whole bunch of other things, and also makes other treatments like even hepatitis B vaccine is much more effective after a BCG. So there's a US trial which came out several days ago, so last week, where type 1 diabetics were given multiple doses of BCG, and they're looking at whether this reduces the amount of like insulin they require to treat their diabetes by decreasing the autoimmune response. Now, coincidentally, this was a few months before the COVID outbreak. So they had this double-blinded controlled trial, and they got their 3 vaccines, and then they looked at what happened with COVID, because the thing was already controlled and everyone got COVID, so you didn't really have to worry about trying to give them COVID deliberately. And it actually produces a 92% reduction in the acquisition of symptomatic COVID. And that's a really strange thing, that a tuberculosis vaccine will reduce your risk of getting COVID, but you do have to do it a long way ahead of time. So I know there was a trial in Australia, I think, where they were giving hospital staff a single BCG, but COVID was already there. And I don't think the trial has produced much information, but this does show that it would actually work. And I guess if COVID became a longer term problem, it may be an interesting sort of idea to increase stop you actually getting symptomatic COVID at all. It also decreases things like secondary melanoma, bladder cancer. It may help with multiple sclerosis, may help with diabetes, as we've already said. Not likely to be reintroduced to our childhood schedule anytime soon, unfortunately, but it would be interesting to see what would happen if people did have a BCG every 10 years or so, in terms of the reduction in death and hospitalization from a whole number of diseases. Tim gets bitten by a dog. Tim's just the unluckiest traveler in the world, I'm afraid. And so he's wandering along in Laos, and there's a dog under a car. It bites him on the ankle. He goes to the capital, which is Vientiane, and he's given equine rabies immunoglobulin, and unfortunately has a severe allergic reaction. 1 week later, he's told, the dog's fine, it didn't have rabies anyway. Now, this is a bit reversed of my traveller. So this was a nursing student. So from Griffin on the Gold Coast, nursing students do a homestay in Laos and do community medicine. And the student was bitten by a dog in the household where she was doing the homestay. The dog was okay. She finished her elective. She then went to Thailand on a holiday after the trip, and then was emailed by the family and said their dog had developed rabies and had to be put down. And there was some mad rushing around by the university, and she did get I think human rabies immunoglobulin and vaccination, and now every nursing student who goes to that Laos trip has to have rabies vaccinations before they go, not for the last 2 years, of course. So equine rabies immunoglobulin is unpurified a lot of the time, and it has a lot of horse antibodies in it. And that's a real problem, because we don't want travellers having to get a treatment which can make them severely unwell when the animal probably never had rabies in the first place. So the other alternative, of course, is pre-travel vaccination. And it's a real dilemma, because public health has to do masses, like thousands and thousands of post-bite treatment on monkey bites. And we're using human rabies immunoglobulin in Australia, which we have to import from the US or Europe or whatever. And we're preventing maybe 1 or 2 bites. I mean, the Balinese claim that monkeys don't get rabies from dogs, but that would seem extremely unlikely. If you're going to give 3 intramuscular vaccines, well, that can cost over $300, and it's hard to tell your family of 4, by the way, can you add another $1,200 to your trip for your budget holiday to Bali, because they don't seem very keen on that idea. But if you look up on the internet, you will find masses of holidays that were terminated because one of the children got bitten by a monkey and the entire family had to fly back to Australia. So what has happened recently is the CDC, the Center for Disease Control in America, has now updated their guidelines, and you only need 2 vaccines 1 week apart before a single trip. And that is both cheaper and also helps us a lot when travelers are leaving very soon, of course. Then they can opt to either have a blood test to prove the vaccine are still producing antibodies, or do a 3rd vaccine for some future trip. And they can delay that 3rd vaccine for up to 3 years. Now, this is not done in Australia. This is the complete guidelines from the CDC, but eventually this would be altered for Australia as well, one would imagine, given the amount of research that's already been done into this. And we're already doing this. I mean, we literally want as many people as possible to have rabies shots, and if we can save money, then we are going to recommend this. This is reliable information. The CDC is a reliable source. And that's the bit about having a teeter in a year, a blood test in a year, or a 3rd dose up to 3 years later. They can do it as little as 3 weeks. It's like the first course. So if they were to do 2 a week before they go, do their 1 week in Bali, come back and do the 3rd one, that is fine. The other thing that has been studied a lot in Australia is using intradermal vaccines, and 2 intradermal vaccines given to each arm on day 0 and 1 week is very similar in effectiveness to intramuscular injection of rabies. And that can reduce the cost by over 50%. But one, you have to be doing a lot of intradermal, and also you have to have several travellers in a day because the vial doesn't last. Once you do one, you throw it away after a few hours. So this is only cheaper if you're doing enough rabies vaccines in travellers to make it worthwhile. But it is, well, we're trying to encourage more people to do it, basically, particularly backpackers spending months or years in Southeast Asia, Africa, South America. And we're starting to see in the last 3 months, a lot of travellers starting to do these sort of quite remote trips because there are less borders to people travelling with COVID vaccine requirements these days. Tim decides to go to London now. Now, London's a very safe place to travel, of course, but what is happening in the world is, unfortunately, polio is coming back, both in the wild form, originally down to just Pakistan and Afghanistan, but now in Malawi and Mozambique, wild polio cases have been reported, came from Pakistan, but now other local people have been infected, but also vaccine-related polio. So if you give lots of babies oral polio vaccine, which is a live vaccine, in some of them it becomes a live vaccine again and becomes infectious. And this is tested for in sewage testing quite regularly in most cities, or most large cities. The problem is there's multiple genetic subtypes. So this is not a single person. They are suspecting there's a whole slew of people around who have polio disease and may be contagious, and they don't know where they are. So in London, they have now recommended every child should get another polio booster. And we're certainly starting to recommend that every traveller should consider now polio again, whereas before we were limiting it only to those sort of 2 countries. So it's a victim of success. I mean, it's been so successful. Most countries have never seen a polio case, no young adults anyway. And of course, COVID meant people weren't going to the doctor as much. And so polio boosters and full vaccine courses have been decreased in less developed countries. And of course, the anti-vax movement may make it absolutely impossible to stop the spread of polio in unvaccinated populations in the future. And we use, obviously, injectable polio, which is an inactivated vaccine. And lastly, diphtheria. Diphtheria you can never eliminate. And again, there are now outbreaks. There was one in Switzerland recently in a refugee camp. And again, these are people who have not been vaccinated at birth, but also there have been a couple of cases in unvaccinated people. And that suddenly means that if you are going to countries again, well, you need now not just like a tetanus shot, but also a diphtheria booster. Now, that's actually not a bad idea then, because when we can do DTPA, we also give them some pertussis protection, and they get the polio as well. It's relatively cheap. That is long lasting. You only need 1 adult polio. So we're starting to recommend this a lot more in travellers who we possibly wouldn't have worried about going to developed countries because they can get a tetanus shot there, but maybe not safe anymore. Lastly, Japanese encephalitis, which Cameron has already talked about, and topical because of those cases. So 1.23 million tourists went to Bali in 2019, but the last Australian diagnosed case was in 2018. He was diagnosed because he died. There have probably been others which have had mild case. He was spending months of each year in rural Bali. The guidelines suggest the vaccine, if you're going to spend a month or more in rural Bali, so not just doing a resort type trip. For children, of course, you've only got Imagev, which is a single vaccine, but quite expensive. So for a family of 4, again, you're talking about another $1,000. So if you want to add their typhoid and Hep A and their rabies shots and their Japanese encephalitis shots, of course, they're not going to be going to Bali anytime soon. And we can prevent the disease. I mean, repellent and covering up should be easy enough to do on a short holiday, because you're also trying to prevent things like dengue fever and diseases of that nature. So these diseases are continuing to come back and attack us over and over again. The population seems to be being brainwashed, believing that the disease is safer than the vaccination. It seems literally insane. So doctor, you can save the world. And of course, we have the nurse in the picture and the helpers and all that sort of stuff. So good luck, everyone. And I love this alliance and trying to give us information that we can give to patients. Like updated information is so important, and trying to tell people that this is why you need it. And we just have to look like we know exactly what we're talking about and present it in a way that makes sense to patients. Thank you.
Thanks very much, Simon. That was a great talk. You really covered a lot of ground there, and you tied in very earlier with the mosquito talk there. So I imagine there'll be lots of questions. We're going to slightly change tack now. And I'd like to introduce Paul Griffin, who's going to talk about new technology in vaccines. Paul was introduced earlier, but those of you who've only joined the afternoon session, Paul is an ID physician in the Mater Hospital in Brisbane, has been involved in the COVID vaccine trials from the start, and is a prolific and informed media commentator who seems to be able to cover all things COVID with alacrity. Paul, I'll hand over to you if you're ready to share your screen, please.
Great, thank you. So just having some minor IT issues. So yeah, thanks to the organisers for asking me to speak. I've got a few disclosures there in that I have run clinical trials for a number of COVID vaccines and do some advisory board and speaking work for a number of relevant companies, but of course the content in this talk is mine. So I'm very quickly, in the 20 minutes I've been allocated, going to just summarise some new technologies to do with vaccines. I've obviously focused on COVID because I think every available platform and technology is being utilised in that area, but this is broadly applicable to a number of different pathogens as well. And so what I'll cover is a bit of talk about variants and a fair bit of talk around the second generation vaccines and why we need them and what's potentially out there. I will cover off some information about the current vaccines as well, just for context, but I won't labour those points. So the slides may look a little bit busy, but I'll go through them fairly quickly. So just to cover off on the mechanisms of action of the current vaccines, I mean, first of all, of course, we've got the mRNA vaccines, and it's not actually new technology, as many people still continue to report. BioNTech has been around since 2008, the company behind the Pfizer vaccine, and Moderna was founded in 2010. And on the right there, you can see the top is a study I did with Moderna on an RSV vaccine in around 2015. And the bottom right is their pipeline. So that technology has not only been applied to COVID, although that's where we've seen the greatest success so far, it's being applied to a large number of other pathogens as well. And again, this audience will be very familiar with this mechanism of action, but basically we're injecting the instructions to make proteins, and there are some specific issues around that. It's a bit unstable, so it does need some kind of coat to keep it stable, and does need to be kept at very cold temperatures. It gives a really good response. It was able to be worked up very quickly for COVID, and is safe overall, with some challenges, including myocarditis. And while we're all familiar with Pfizer and Moderna, there are a number of other vaccines that use this platform, and there are also DNA vaccines, and I'll come back to some of those because they've actually been applied more to alternate delivery methods. So we've got some skin vaccines that are DNA-based, and also an oral vaccine, and I'll go through that in a bit more detail as well. Interestingly, viral vector vaccines, whilst used prominently for COVID, are also not new. So we've had viruses as vectors since the 70s, adenoviral vectors since the 80s, and then we had some other adenoviral vector vaccines, including for things like HIV and Ebola. And the Oxford group who made the AstraZeneca vaccine, they were driven by Ebola some time ago. And like so many of the groups that came up with these vaccines, had a lengthy program preparing to respond to so-called Disease X. So they developed their ChAdOx1 platform, which was essentially a plug-and-play viral vector vaccine. And the interesting thing about these vaccines being essentially plug-and-play, meaning that you really only need the genetic sequence of the pathogen, does mean that not only were they able to be established relatively quickly, they can also be updated relatively quickly. And I'll come back to that a little bit later. And again, I think most people would be familiar with this, that the gene for the spike protein is inserted in a benign, unrelated virus, delivers it in a way that's functional and enables the spike protein to be made and for an immune response to be given against that. Importantly, that these are replication deficient, so they don't reproduce. And that's one area where some of the updated vaccines are a little bit different and might be able to provide an even greater response by doing that. And I'll come back to that. So, of course, we have used the Oxford AstraZeneca vaccine quite extensively in our country, but there are some other options, including the J&J vaccine, which was notable as it was a single dose. And protein vaccines is where perhaps a more traditional approach was used. We had the recombinant Hep B vaccine in the 80s, and the HPV vaccine first made in the 90s and approved in 2006. But for COVID, the first one here was Novavix, which is not a new company, founded some time ago. They had vaccines for other pathogens, including the other coronaviruses of SARS-CoV and MERS, a lot of work on Ebola. But hadn't commercialised a vaccine pre-COVID, not because there were any issues with their vaccines, I think just didn't get to that point. And I'd done a lot of work with them before, including on their RSV vaccines in healthy volunteers, elderly, in pregnant women, as well as flu, including some novel flus like H7N9, and a fair bit of work on their Ebola vaccine. And you can see from the top right, they also have a fairly lengthy pipeline where they're looking at a number of other pathogens to apply their technology to. And so this was the first vaccine trial in the Southern Hemisphere, generated great results, published in the New England and approved for use in our country. Took a lot longer for them to get through those trials, not for any safety reasons, but for some logistical ones, and is now approved both for primary vaccination as well as a booster in Australia, although there's been some challenging recommendations recently, and it doesn't seem it's being as strongly recommended as a booster as some other vaccines when it was actually approved to be used in that context. And again, a very simple mechanism of action that many people would understand. So you just make the target antigen in the lab, often don't get a strong enough response without doing something else to boost that. So we usually have an adjuvant, for example, and some other technology, nanoparticles, which is what Novavix do to assemble it so it looks a bit like the virus, and things like the molecular clamp and the UQ vaccine, which I'll briefly touch on in a moment. And while the Novavax is the one we've heard the most about, Sanofi also trialled their vaccine in Australia and have some regulatory approvals underway, and there are a host of others as well. And finally, in terms of the COVID vaccines, we've had inactivated and attenuated, where basically we just kill the virus and use that. Most commonly, it's been using beta-propiolactone, which binds to the gene so the virus can't replicate, but it still kind of looks the same. And this has been used in a number of countries without the same success, I think, as the other platforms. But there are some that are used elsewhere that are recognised for travel-related reasons when that was really an issue in Australia, but not approved for actual use here. So I guess the first improvement on our current vaccines is variant-specific boosters. So I'll quickly touch on that. So in terms of the background, we know that variants are arising. It's kind of a random event that these viruses just change, but when it confers some kind of benefit, and you can see that can be to do with transmissibility, severity, or it can mean an improved ability to evade our countermeasures like testing or vaccines, and it's called a variant of concern. And we've seen, obviously, the emergence of these fairly rapidly, and they're going to continue to emerge. We do need a strategy to address, and that strategy is the variant-specific booster. And as I mentioned before, these are nearly all plug-and-play type platforms, so it's pretty easy to change. A lot of work was done to have an established platform to be able to update them, just really using neutralizing antibody studies in the lab and maybe some clinical trials to supplement that. And a lot of talk before this was actually done that it was going to be around 8 to 10 weeks from discovery of a new variant that necessitated this change to rolling that out. And we actually had variant-specific boosters developed for alpha, beta, gamma, and delta. But when they were tested, they offered no advantage over the original vaccine, so didn't need to be deployed. But obviously, Omicron really changed that very significantly. And again, a lot of detail here just to frame how significant Omicron is. I won't go through this in great detail. It seems like we've been dealing with Omicron for a long time, but it was really only first reported the 24th of November last year. Lots of changes, both in the genetic sequence and then in its properties. We then saw BA2 emerge from the Philippines, more infectious once again. And then we obviously saw BA4 and BA5, and these were more recent again, 10th of January or the 25th of February, respectively, and had much greater immune evasion potential. And the vaccine still provided good protection from severe disease, it's important to note, but we didn't see the same sort of protection that we wanted, particularly from 2 doses. And obviously people will be familiar with the extensive discussion around Australia's so-called third Omicron wave that we're hopefully just on the tail end of at the moment. And so Pfizer and Moderna, as well as many other countries, have been developing these Omicron-specific boosters, and Novavax is still trialling theirs in Australia here at the moment, and they're mostly bivalent. So not only are they targeting Omicron, but they're including the original Wuhan strain as well. And there's been seen to be some advantages in doing that. And some of the figures quoted with neutralizing antibodies to BA1, Pfizer, probably a 2-fold increase, Moderna not far behind. And while that was to BA1, there's also been improved protection from BA4 and BA5, although not quite to the same degree. And when preparing these sort of talks, you do have to update these slides quite often because the MHRA approved Moderna's Omicron-specific booster just last week. And the provisional determinations are in with the TGA for both the Moderna and the Pfizer vaccine, meaning that they're looking at the data and it shouldn't be too far away. A lot of people are asking exactly when and should they, excuse me, defer their scheduled booster now. And the simple answer there is I would not encourage waiting because until they're approved and available, we simply don't know exactly when that will happen. So moving on from the first step forward, those Omicron-specific boosters, is to so-called second-generation vaccines. And again, I think it's important to note that this doesn't detract from both the safety and efficacy of our current vaccines, but it's clear their properties aren't perfect. And so in terms of the additional desired properties we'd like, and there's probably no one vaccine that's going to satisfy all of these, so it's likely to end up being combinations we might look at. But single-dose vaccines, long-lasting, maybe lifelong protection, high efficacy in all outcomes. So we know a lot of discussion with current vaccines about severe disease, but it'd be great to see something that blocks transmission and both infection and onward transmission, a vaccine free of side effects, thermal stability, maybe room temperature, long-term stability, so we can ship them around the world, easy and cheaper to make, for example, and being able to give in by an alternate means other than needle and syringe. So these are all things that are being worked on. And when I looked at the tracker about 6 months ago, this is where we're up to, about 115 vaccines in clinical trials. And you can see just in 6 months, that's jumped up. And if we look at some sources, there's reported around 120 vaccines in clinical trials. You can see this number adds up to more because some are in multiple phases simultaneously. And then again, not expecting people to be able to read this detail, but this is the WHO tracker and landscape where they report there's some 170 vaccines in various stages of clinical development. So it remains a very active area. And so just in terms of some of the vaccines that I've been involved in here in Australia, we obviously had the UQ vaccine, which was the molecular clamp, and that you can see the pictures at the bottom. Sometimes these vaccines change, they fold or bind together into a configuration that's not the same as the antigen that we want to get protection against. So some clever people at the University of Queensland added this molecular clamp to keep things in the right shape. Unfortunately, they chose something that led to some diagnostic interference, so their COVID candidate didn't proceed, but I'm confident this is technology that we will see be used maybe against other pathogens, hopefully in the not too distant future. In terms of greater specificity and reduced susceptibility to change from new variants, one approach is to not use the whole spike protein, but to look at more specific areas, and a very active area is the so-called receptor binding domain. So the bit the virus actually uses to bind to our receptors, the ACE2, and there's lots of work underway. One that we were involved in here is a Serum Institute of India study with some clever technology where they actually attached it to the Hep B surface antigen, again, to hopefully get a better response. And this is one that's still ongoing, progressing fairly slowly, which may not be a good sign. An oral vaccine was a really exciting one that I was involved in. So a bit like a viral vector approach, but instead of using a harmless virus to carry the payload or the genetic material for the spike protein, in this case, they used a harmless bacteria, essentially a probiotic. And the idea here was a number of benefits would arise from this. Given orally was really important, room temperature stability, but also hopefully given it was being administered via the mucosal route, maybe a better mucosal response. So a very promising technology that again is progressing a little bit slowly through clinical trials, but we'll keep a close eye on that one as well. And then perhaps the most active area, and I think the most discussed in recent times, is intranasal vaccines. And this company, we've done their clinical trials. And so a little bit like a viral vector vaccine, but instead of it being replication deficient, this one can undergo a single cycle of replication. So it reproduces once, and that's thought to give about 100 times the amount of spike protein produced. And so I'll talk a bit about the pros and cons of intranasal approach in a moment, but overcoming the difficulty getting a strong enough response might be able to be done by this approach. And we started off using this intramuscularly, and now have done a number of cohorts intranasally, and that work is underway. And just to point out as well, the Sanofi group, they had both an mRNA and a recombinant protein vaccine. The mRNA wasn't taken forward, but their recombinant protein before the variant-specific boosters was actually a bivalent approach as well, which, similar to why we're using that with the Omicron-specific boosters, is thought to maybe give broader cross protection. So the one approach to hopefully improve the situation there. And where there is a lot of interest is in these novel delivery methods. And so something that could be given without a needle and syringe is actually a really important step. And so a lot of people are needle phobic, but needle and syringe administration does come with a lot of other challenges, including sharps disposal and how they're administered, for example, and maybe even some challenges in terms of immunogenicity. So there are a lot of different ways of trying to give vaccines that are being looked at. In terms of the skin, I mentioned that DNA vaccines are an active area, and a number of these groups have had vaccines for other pathogens before. They often need some kind of clever way of actually trying to get the antigen in. You can see the picture on the right is some kind of injector, and some of them even use little electric impulses to try and deliver the DNA through the skin. So a bit more of a complicated approach, and that might be something that's a bit of a challenge. But again, another clever University of Queensland group came up with this so-called Nanopatch, which is like having lots of tiny micro projections on a little square-shaped device. It's about 1 centimetre by 1 centimetre, and that's put on with a fairly simple administrator. The picture there is of a slightly older version. It's now a little bit more compact and easy to use again. And you just hold that on the skin and push it down, and it enables those micro projections to be put into the skin. And there's a lot of potential benefits other than it just being not needle and syringe, in that it may actually give a better response given the local immune cells present in the skin as well. And as I mentioned, intranasal is a very promising approach. And the main reason here is it's thought that giving the vaccine via the same route through which the virus gets in might be better at developing a transmission blocking response, or at least some kind of mucosal immunity that might be better at doing that. And depending on the source, it appears there's between 8 to 12 intranasal vaccines in various stages of clinical trials. But it's not as simple and easy as it sounds. We do see limited responses with some of them, and people might recall the story with flu mist, where we had intranasal flu vaccines that then were shown to not really be doing that much. It's a bit hard to administer via this route. People can sneeze, or if they don't hold their head right or squirt it in properly, they might not get the full dose. And so there are some challenges, but as I say, the transmission blocking potential here is a really exciting one. And what's looking most likely, and supported by some really good animal data recently, is it might be what's being called a prime and spike approach. So maybe we prime people with intramuscular vaccines we're already using that have been shown to maybe generate some mucosal immunity, but it's pretty poor, to be honest. But then boosting with some kind of mucosal vaccine, like an intranasal one, to give both systemic protection and particularly that severe disease approach, but also something that will stop the virus getting in. And as I mentioned, there's quite a few of those around. The Tetherx vaccine that we're looking at, Oxford AstraZeneca are using their approach, and there are quite a few others in various stages of clinical trials. Some of the trackers would indicate there may be 3 that are in phase 3 trials, but it's hard to get a lot of really good information around that at the moment. And here's the thought, is that there's a lot of different approaches, and what we might see is we get better protection from infection, better prevention of lung infection, clearing the infectious virus in vital organs, being able to clear the virus from the upper respiratory tract so people are no longer infectious as well, and preventing viral shedding and transmission. So, this would be something that would obviously be tremendous if we actually were able to get that to work. And of course, an oral vaccine, perhaps even more challenging, but maybe even more beneficial. And there are a number of different approaches there, and maybe the mucosal situation might be relevant to that as well. And just quickly to wrap up, talking about variants moving forward, obviously we know that new variants are going to continue to arise, so we do need a better approach so that we're not always, if we try and just chase our tail and adapt to a variant-specific booster, given the rate of evolution of those, we're probably going to be falling further and further behind. So variant-proof vaccines or a pan or universal coronavirus vaccine is what's being looked at. And I've listed some of the approaches there. An interesting one, I think, is that last one with ferritin nanoparticle with Walter Reed. So this is a particle that's got 24 sides. And the thought there is if they basically saturate that with different parts of the virus, again, broad across protection, and maybe even used more conserved regions, so things other than the spike protein in addition to the spike, to get a broader cross protection response as well. Combination vaccines, and I think I'm getting close to time, but it would be great to make it easier again to administer vaccines. And we saw all the challenges with flu. We kept it away for a long time. It was clearly back this year. Some challenging recommendations about initially separating COVID and flu vaccines, but it was clear you could give them at the same time. And now there's a lot of combination vaccines in fairly mature stages of clinical trials, Novavix, Mrna, etc. And Moderna's candidate even has COVID, flu, and RSV combined. Of course, one of the challenges we always worry about with combining vaccines is it might reduce protection against one component or possibly all of them, but it's looking really promising that that's maybe not the case. I think the biggest challenge moving forward for us will be the fact that COVID isn't going to be necessarily seasonal. So when would we use these vaccines? But if we go to a less frequent boosting option, maybe a combined vaccine might be something that we can use. So just to quickly wrap up, so obviously we've been extremely fortunate to develop the vaccines that we've used, and the fact that there's ongoing work in this space doesn't mean that we shouldn't be very happy with how safe and effective they are. And we're now using those from 6 months of age upward, which I think is fantastic. But we do need some improved vaccines, I think, to supplement what we've got already, including the main one, I think, which would be an intranasal transmission blocking vaccine. That boost and spike approach would just be amazing. And I think when we put this into context, we know COVID-19 is here to stay, but we are getting better tools all the time. So we're going to get variant-specific boosters this year, most likely in our country. We'll probably have some second-generation vaccines next year to improve the situation. There's not going to be a silver bullet, though. Our therapies are tremendous, with some logistical challenges to overcome there, but hopefully we'll expand their accessibility. We've got prophylactic and therapeutic antibodies, again, for people who may not be able to respond to vaccines. So again, a broad combination approach to using those tools. I think it's reasonable to expect a high level of control moving forward. And I'll leave it there.
Paul, you've done extraordinarily well, and it's almost we should get a diploma from the things that you've covered if we can pass the test later. We have one more speaker in this session, and we're going to take a deviation from infectious disease, and we're going to be looking at vaccines and cancer. So I'm very pleased to introduce Professor Kristen Radford, and she leads the Cancer Immunotherapies Group at the Meta Research at the University of Queensland in the Translational Research Institute. She's had a background in developing a dendritic cell vaccine, which was the first clinical trial for metastatic prostate cancer. And she's ideally placed to talk to us about the emergence of vaccines in either preventing or treating cancer. Kristen, could you please share your screen, and I'll hand the podium over to you.
So I'm joining you today from the Translational Research Institute in Brisbane. We're on the Turrbal and Jagera people's lands, and I pay my deepest respect to their people, their culture, and their elders past, present, and emerging. And so I guess the concept of using the immune system to recognise and treat cancer has really been around since the 19th century. It's not something new, but it's really only in the last decade or so that we've actually seen the immune system to be a benefit to cancer patients and in terms of treating the disease. And on this slide is just an article that's a little bit old now, but it was on the cover of Time magazine, just showing that, really emphasising the point that the immune system can be used as a treatment to recognise and fight cancer. We've got a number of challenges to overcome, but it's really, at the moment, hugely expensive, but can be transformative to some cancer patients. And really, there's three main types of approaches to harnessing the immune system for cancer therapy. The first is what we call the immune checkpoint inhibitors, and you may have already heard of those before. These are now being used as first-line treatments for a variety of cancers, but they've really been most effective in melanoma and lung cancer and others. And what they're really designed to do is, if you've already got a pre-existing immune response against your cancer, these are designed to turn the brakes off that are either slowing down or impairing that immune system, and be able to ramp that up so that the immune system can recognise and kill the cancer. And as effective and transformative as they've been in the last 10 years or so, they're really still only effective in about 10 to 15% of cancer patients. And so I guess one of the real challenges in the field is how can we make these treatments more effective? On a lesser scale, they're around about $150,000 to $200,000 per patient per year, those. On a smaller scale, but a very emerging and rapidly developing field is what we call cell therapies. And these are taking components, and particularly the CD8 or killer T cells of the immune system, genetically manipulating those and infusing those back into the patients where they can recognise and fight cancers. These have been very, very effective in leukemias. And again, don't come cheap at a price tag of around $600,000 to $700,000 per patient. And then the third approach is the cancer vaccines, which I'd like to focus on a little bit more today, because that's more my area. So these are a couple of sort of schematics of what cancers look like. And on the left, we've got a cold tumour, and that's what probably 80 to 90% of cancer patients, and this is very generic across all different types of cancers, actually look like. We don't see very many immune cells coming into there, and the ones that we do see that come into most cancer patients are what we call suppressive. So they're actually keeping the immune system quiet, dampening it down. And so these are the real challenge, and these are the types of tumours that we think may be beneficial of a vaccine approach that could really induce and ramp up immune responses. On the right is what we've got, what we call a hot tumour. And so those little gold balls with the red rings around them are really these CD8 killer T cells and other different types of immune cells. And what happens in very few patients is that immune infiltrates are quite often associated with better prognosis for cancer patients. They are also associated with better responses to these immune therapies. So those checkpoint inhibitors, for example, work best where you've got a hot tumour, it's got lots of killer T cells in there already, and those sorts of drugs are really designed to ramp up and activate those killer T cells. And just a little bit of a spoiler, I guess, is the little gold stars. They're called the dendritic cells, and they're a rare immune cell type that's really essential for initiating and directing all those immune responses. They're the cell type that's my particular area of interest, and they're one of the aspects that we think might be the key to more effective vaccines. And so what we're trying to do with cancer vaccine is really to try and induce or establish some kind of long-lasting immune response that can be used either to treat an existing tumour, or in a patient that's already had a cancer and is already in remission, to prevent that cancer from coming back, or to prevent resistance against other diseases. And of course, like all vaccines, we really want to avoid non-specific responses or any kind of adverse effects. And that's particularly challenging for cancers because cancers evolve from your own body. And so the immune system sees that response as self. It doesn't see it as foreign as it would depict a pathogen. There's two main types of cancer vaccines. So we've got the prophylactic vaccines. These are really designed to prevent infection of a pathogen that is known to increase risk of particular cancers. And of course, the typical examples we have there are the human papilloma virus vaccines such as Gardasil and Ceravax that prevent cervical cancer, and hepatitis B vaccines that can prevent hepatocellular cancer. So I'm not going to talk too much about those today because they're really very similar principles to vaccines that you've heard a lot about already today, and they're really about prevention of that viral infection as opposed to treatment. What I'd like to spend a bit more time talking about is the therapeutic vaccines. And what we're trying to aim to do there is really treat a cancer that's already in existence. So we do not have those same sorts of foreign antigens that we would from a pathogen or from a cancer that is caused by a virus. And so that presents itself with particular challenges. A couple of examples that are already FDA approved, though, is one called Sipuleucel T, and that's a dendritic cell or a cell-based therapy vaccine that's been approved for metastatic prostate cancer. As we've heard earlier, there's the BCG vaccine, which is also an FDA-approved treatment for bladder cancer. So one of the first challenges, I guess, is the type of immune response we're trying to generate or really enhance in terms for cancer vaccines is very different from the type of immune response that we're trying to generate against a pathogen. So most of our vaccines against pathogens are really the main focus, or the primary endpoint is really about inducing neutralizing antibodies in the first instance. Tumor immune responses are largely mediated by the CD8 cytotoxic or killer T cells. And so that's really the primary type of response that we're aiming to induce with a cancer vaccine. And those killer T cells are educated by dendritic cells, and these are the sentinels or professional antigen presenting cells of the immune system. They take up the antigens and bits and pieces from pathogens and from cancers and process those in a format that those T cells can be recognised and activated and expand. And so it's really those key interactions. One of the key challenges that we really have is making sure that we've got a vaccine platform that can really highly specifically deliver the antigen to those dendritic cells to be able to induce that killer T cell response. And that primarily happens in the lymph node. So there's some additional challenges that we've got when we're trying to look at cancer vaccines. And really the first one is that every patient's cancer is different. Even within the same tumour mass, most of the time there's a lot of the cells that's very heterogeneous, and so there's different mutations and different cell types even within a single tumour mass, so that's particularly challenging. One cancer patient's tumour is very different to the next one as well. And even within a cancer patient, one tumour mass can even be very different to another tumour mass in a secondary location. And so this all presents some really key challenges in terms of what are the antigens, or how do we get the immune system to recognise a cancer, and what are the particular determinants that we can use to recognise that. And of course, there's additional challenges on that in that tumours evolve and mutate to avoid the immune system. And so particularly over time, with various treatments, for example, those tumours will evolve and display different antigenic determinants, which makes finding a target that the immune system can recognise rather challenging. One of the ways that we can avoid this is an emerging concept that we call in situ vaccination. And this is being trialled with a number of different viruses at the moment. There's lots of clinical trials, but to date, there's only one that's been FDA approved, and that's this T-VEC vaccine that's approved for malignant melanoma. And what that is it's an oncolytic virus. So you inject that virus directly into the tumour mass, and that virus then infects the tumour cells and induces lysis of those. And so that releases those tumour determinants and a lot of inflammation that brings the rest of the immune system in, that can then be activated to induce a systemic response. And so there's a number of advantages of that. Obviously, you don't need to find what those cancer antigens or determinants are that the immune system can recognise, because it finds that for itself. One of the main problems with this is that you need an accessible tumour. And so this type of approach at the moment is largely limited to cutaneous or accessible malignancies where you can inject that virus in. And of course, it's even though these viruses have tropism towards the tumour, they're not necessarily selective for the tumour as well. So there are potentials for off-target effects, but that's one way we can get around it. A more targeted approach is really trying to work out what those actual cancer antigens are, what the immune system can recognise. And what we have here is two main classes of tumour antigens. And so the first ones are what we call self-antigens. And so they're proteins that might be upregulated or overexpressed in the cancer relative to other tissues. So they make good targets, because obviously you don't want your cancer target to be expressed in the immune system to attack your healthy tissues. The caveat of those types of antigens, though, is that by natural process, your immune system's already seen those. And so the really highly reactive immune cells, T cells, have been eliminated or selected out. And so that means that there's generally weaker responses against those types of antigens. It also means that autoimmune toxicity is possible if those antigens are expressed in other tissue types. But examples where these things have been used successfully, for example, are proteins that might be expressed in melanocytes, for example, that the side effect might be vitiligo, which is then not necessarily threatening, or things like prostate-specific antigens for prostate cancer. The advantages of those types of approaches, though, is is that they're more broadly expressed across different tumour types and against a more broader range of cancer patients. And so what that means is that you could potentially make a generic vaccine. It doesn't necessarily need to be patient specific. It also means then that vaccine can be generated in an off-the-shelf manner, so it's ready for when the patient's diagnosed and as soon as they need it. The emerging concept in the field now is what we call neoantigens. These arise because particularly certain types of tumours such as melanoma and lung cancer have a high degree of mutational load in them, and that varies over time. So the more mutations that a cancer generates, these are specific to the cancer, those mutations aren't expressed in the healthy tissue. And those mutations essentially give the immune system more things to be able to see and recognise. And so they're in essence, they're truly foreign proteins, and that makes them much more immunogenic and a larger degree of potential success because you've got a larger number of them. The potential for autoimmune toxicity is much lower for those because they're not expressed in the healthy tissues. The real problem with this approach is that all those mutations quite often very patient specific. And so these types of approaches need to be very personalised and tailored towards specific patients. And then the caveat of that is that you need to custom make those. And so you need to wait till after the patient's being diagnosed and, then you've got, depending on the vaccine platform, various periods of time before the patients can actually access that vaccine, which may be detrimental for them. And this is just a study that was published a couple of years ago that shows the burden, the tumour mutations. So some types of cancers, particularly melanomas and lung cancers, have higher levels of mutations. And those types of tumours are known to have better immune infiltrates and be more responsive to the checkpoint inhibitors and immunotherapies. And so that's really good evidence that some of these cancers with really high mutational loads may be really amenable to, particularly to these sorts of neoantigen type immunotherapies. Whereas other tumours that have really lower mutational burdens, and particularly down the bottom end of things like ovarian and prostate cancer and even pancreatic cancer, less mutations, so less things for the immune system to see. And so they may be more amenable to or more restricted to using some of those shared antigens. And so this concept of personalised cancer vaccines and neoantigens is really starting to take off. So the approach of how you do this, it's really started to come to the forefront with being able to do genomic sequencing on every cancer patient at a more affordable price, I guess. And so the first thing you need to do is get a tumour biopsy and do that DNA sequencing. And there's a whole bunch of various computer algorithms now that can pick up those mutations and then feed those mutations through a bunch of algorithms to predict whether the immune system is likely to recognise those or not. From those, you take the highest hits, and generally that's about 20 to 30 potential antigens, and they can be made into various vaccine formulations where they're then taken into the patient. And so this is a really highly personalised, tailored approach. So, it's predicted to be more effective. The problem is in this approach at the moment, they're taking about an average of 4 months to get this process for individualised patients. And usually by the time patients, many of them get to this stage, that's too late. They don't want to be waiting 4 months or the potential of even passed away before their vaccine's ready. And so getting this down, making these were coming in at just under $1 million per patient. So getting these into a more rapid turnaround and a more affordable price bracket is where we need to go in the future. And so the other thing I really wanted to talk about in terms of consideration, and Paul already talked about this before, is the various vaccine platforms that we can use. And pretty much all of the different platforms that have been used for infectious disease have also been trialled. in cancer, and that includes peptides or proteins injected with adjuvants, various different forms of viral vectors and DNA vaccines, and even killed or genetically modified tumour cells have been injected in. Because I'm a dendritic cell expert and a bit dendritic cell centric, and they're so essential for this priming of the response that we need, we kind of classify those as randomly targeting dendritic cells, because in essence, those things have got to get in, they've got to not be degraded and find their way to the dendritic cells and the lymph nodes before an immune response can be generated. And so to overcome that, particularly because of these weaker T cell responses that we generally see against a tumour in comparison to a pathogen, one approach that's been looked at for the last 20 years or so has been to take the dendritic cells out of the patient's blood and instruct those in vitro. So we load those with antigens and various adjuvants and reinfuse those back in. Moving towards the future, and some of the vaccines that we're looking at developing are trying to overcome that again and combine the peptides or the potentially neoadjuvant type approaches, vaccine platforms, but combine those with much more improved specificity so that they're very specifically targeting those dendritic cells in vivo. And I think ultimately what we need is we need to be able to combine these with other different therapies, particularly treatments that can overcome immune suppression or that can reactivate the immune system, such as those checkpoint inhibitors. And so I'll just talk a little bit about these different approaches in a little bit more detail on where we are with those. And so the mechanisms of action, we've already heard a little bit about how DNA and RNA vaccines get in. They need to be transcribed into a protein and then presented so that the dendritic cell can be recognised. All of these have been used in various clinical trials in a whole bunch of different cancer patients. The other caveat with cancer patients is that all of these are generally tried in late stage patients who have failed all other therapies. They're very small trials as well. And so getting enough patients in a rapid enough time to actually get these things moving and demonstrate some efficacy is a particular challenge. But these sorts of platforms, so DNA, RNA, protein peptide based vaccines, we've got a lot of experience already from the infectious disease vaccine world that can be easily applied and has been applied using these different technologies. They have the advantage that we can manufacture them. We can use either those shared antigens or personalized antigens, but that takes a bit of time. The big problem with these really is their stability. So it's a bit like delivering a fragile package and just dumping it in your suburb and hoping that you'll come and find it. Because it doesn't get specifically delivered to the dendritic cells that really need it. And so particularly protein peptide vaccines, you get preferential CD4 T cell responses, which are important, but they're really not the main effector mechanism that we need to fight cancer with. And so getting enhanced delivery platforms that can advance those CD8 T cell responses is really important. I just wanted to touch briefly on RNA vaccine for cancer. If we didn't have these already done in the last 5 years or so, this is really underpinning how we're able to move so quickly with the RNA vaccines for COVID, because these had already, the platform had already been established and undergone phase 1 and 1/2 trials for metastatic cancer patients. At the moment, there's over 20 clinical trials in various phases. They're using different combinations. So some are just making these shared tumour antigens into RNA and putting them in. Others are going through that whole process of identifying patient-specific antigens and then formulating those into an RNA vaccine formulation, injecting them in. There's a whole bunch of different cancer types being trialled here, and all the big players have skin in this game. At the moment, they're really showing in these early trials that they're safe, they're tolerable. There's some evidence of immune responses and efficacy, but again, these are still fairly early days for these types of approaches. The other type of approach is the dendritic cell therapy. So this has been around for over 20 years. There have been thousands of cancer patients that have received these, and again, mostly in early stage trials. And so this idea is taking the immune precursors, the antigen presenting cells, out of the patient's blood and manipulating those in the lab and then reinfusing those back in. There's no doubt
we're running out of time. We're running out of time.
Okay, sorry, on overtime. So there's no doubt that they're safe and well tolerated as well. There's one of those that is already FDA approved, and that's Progen for metastatic prostate cancer. Because of these logistics of making cell-based therapies, they're not going to be particularly widespread use. And another one to watch for is this Rna Vax, which is in phase 3 for glioblastoma. If I've got one more minute, I'd really like to just talk to you about the new approaches that we're using. And these are using antibodies specific to dendritic cell receptors to deliver protein-based vaccines and hopefully eventually RNA vaccines. And so this gives us much more specific targeting. These have already also been looked at in very early phase clinical trials and demonstrated safety and some evidence of immune responses. The one we're particularly interested in is what we have discovered is that there's one particular type of dendritic cell that we call these DC1s. And they've been really shown to be absolutely critical for tumour immune responses and for CD8 T cell immunity. And so what we believe is really that developing the next generation of cancer vaccines will involve antibodies that specifically target this one particular key subtype of dendritic cell in vivo. And of course, that enhanced delivery to CD8 T cells will give us stronger killer T cell responses with lower potential for off-target effects compared to the conventional vaccines. I'll just skip over this very, very quickly, but this is some evidence from our lab in our prototype vaccine that shows that indeed this will probably be the case. So just to leave it there, I think therapeutic vaccines have real potential. There's a lot of issues and caveats that we still have to work through. They're largely mediated by these DC1s and the killer T cells. They can be applied to a number of different cancer types and potentially combined with other treatments. And really improving the efficacy by delivering to these DCs in vivo has real potential to improve these responses, as is more patient-specific personalised vaccines. So thank you very much.
Kristen, that was a tour de force. I think we could have had a whole other day on cancer vaccines, and it's kind of something which we should be thinking about for annual scientific meeting in February, but that was a real tour de force on the area there. We don't have that many questions in the chat, and Paul's having to do a radio interview, so we might have to, if there's any questions for him, we might have to hold them. I have two quick questions to kick off, and then I'll perhaps ask any of the speakers if there are any questions that they might have of the other speakers as well. Can we start with Cameron, the mosquitoes? I can say from our personal experience, we live in the Adelaide Hills and we've been here a long time, and the mosquitoes are now around in winter when they were never there before. And I noticed you sort of talked about critical temperatures, but I was just thinking it's they seem to be adapting. So I understand about the tropical side of things, but can you comment on the other side of it? Because we would get mosquitoes in the summer because we have a septic tank, but we're now actually seeing them throughout the year. So I don't know what's actually happening with that.
Yeah, that's a good observation, John. I'll give you an honorary entomologist badge for that question, because it's a good observation, because we're finding the same here in New South Wales, is that while a lot of the discussion about a changing climate is about new types of mosquitoes coming into Australia, I think we are now acknowledging it is that an extension of the mosquito season is something that we need to be mindful of. And even my formal mosquito surveillance programs are the same. There was a time 20 years ago where I wouldn't really start monitoring mosquito populations around Sydney until mid to late November. But now we find mosquito populations are around in September, and they can persist all the way through to May as well, depending on the prevailing climatic conditions. And I think one of the really interesting things we've seen in recent years is that while it's the maximum temperatures that might be ever increasing that we notice a lot, it's the increasing mean minimum temperatures during autumn and spring that really drives a lot of mosquito populations, because it doesn't get cold enough to stunt the population growth. And so I think that into the future, we do need to be mindful about, some of this mosquito activity that's, early and late, because it not only has an implication for potential exposure to mosquito-borne pathogens, but it has an operational implication for those of us who are managing state and territory mosquito and arbovirus surveillance programs. We need to start a bit earlier and be prepared to run a bit later than what we might historically have done.
Can I throw in a quick question, John?
Please do.
Just because we've got Cameron there. I'm completely intrigued by this concept of wind-blown mosquitoes travelling thousands of miles. Can they actually do that?
It seems ridiculous, doesn't it? We know mosquitoes vary quite a lot on how far they actively disperse or fly from their what we call their larval habitats, the wetlands or water-filled containers where they complete their immature development. Some of the dengue mosquitoes, for instance, might only travel only a couple of 100 meters. Some of the Australian wetland mosquitoes travel up to 20 kilometers. We even mark mosquitoes, release them, catch them again, and then found them traveling about 5 kilometers. But there's strong evidence that there are mosquitoes and other biting insects that move from Indonesia, Papua New Guinea, that region into northern Australia, that are dispersed by storm events like cyclones. And we can see that not only from the detection of new mosquito species, but also from outbreaks of other arboviruses. And so our colleagues that work in veterinary health have seen outbreaks of some of the wildlife diseases that we suspect are due to biting midges being redispersed by some of these sort of cyclones and other storm movements. So it seems ridiculous, but mosquitoes are pretty hardy, even though they might appear quite fragile. And it's most likely that is, potentially how some of these pathogens can make their way into Northern Australia.
Interesting.
Back to you, John.
Simon, I have a question for you. As a child who ended up getting the BCG as we all lined up in grade 7, and also as a GP, being mindful of previously having done nursing home visits, that older people, even if they've had a BCG, can have a reactivation. They're less likely. Your comments about a BCG in terms of TB areas, what advice would you give to the folk that the cruise folk, the people that are travelling that are older, that might have either had a BCG at a very young age, or are probably, if they haven't ever had a BCG, are probably at increased risk from TB as they get older?
Well, the risk isn't great just on cruise boats. Effectively, you have to live in houses. It requires quite a bit of coughing. It's not a one cough disease, although there have been cases where people have sat beside someone on an 18-hour plane flight have managed to acquire TB. But your short-term travelers are not really in indoor contact for long enough in general to acquire TB. But for older travelers who want to do volunteer work for a year in a risk area, that is the question. Should they receive BCG? Now, a lot of countries, they don't get one BCG, they get 2. They get one at birth, and then they get another one at high school to maintain their immunity, because they're being exposed all the time, of course. So whether you do MAN 2 testing on them first to see if they're going to react, in which case you probably shouldn't give another BCG, is the issue, I guess.
Can I do the supplementary, John?
Absolutely.
Fantastic talk again, Simon. Typhoid completely fascinates me. Like TB, it was a disease of the 19th century and into the 20th century. You talked about all those vaccines, including the one that put me in bed in 1986. Yeah, but I've been reading about typhoid conjugate vaccines, and I wasn't sure if you knew much about those and their application.
No, because they haven't been released in Australia, and I haven't seen the CDC data on them. It'd be absolutely fantastic to get other typhoid vaccine candidates in Australia. But our real problem is we can't even get the oral vaccine at all at the moment. There's been just shortages of a lot of the vaccines. So we hope as more and more people are traveling, we're going to be able to get those sort of supply chains running again. But obviously 70% is not good enough. We would love to have a vaccine that's 95% effective. Hopefully we will get that in the future.
We're almost out of time. We've decided to cut the discussion session a bit briefer, and be starting the next session, I think, in about 15 minutes, so that will be clarified. So thanks, everybody, and we'll look forward to the next session.